Hello, everyone, and welcome to the session titled Xenotransplantation, When Pigs Fly. Thank you, everyone, for joining today. We do have an expert panel, including one virtual as well, so you'll be able to see her on the screen when she's speaking. My name is Mike Mendicino, and I'm the Chief Consultant of Hybrid Concepts International, which is a cell and gene, regen, med, and xeno-focused consulting firm. And we've been in business about 10 years. And on a personal note, I did my Ph.D. in xenotransplantation, and my first job out of the gate was for a xenotransplantation company. So while I haven't worked in xeno for a long time, it's reemerged, and I've been enjoying seeing its evolution and progress of late. Before I get into a 10-minute introductory presentation, I'd like to pass it to our expert panelists for their introduction. Heather, can you please go ahead?
Hi, Mike, thanks, and good afternoon, everyone. Hi, my name's Heather Lombardi, and I am the Director of Regulatory Strategy at United Therapeutics Corporation. Prior to joining UT, I was actually with FDA for about 17 years, where I held various leadership roles, both at the Center for Veterinary Medicine and the Animal Biotechnology Group, as well as the Center for Biologics at CBER's Office of Therapeutic Products, where I was the director of the Office of Cellular Therapy and Human Tissues. Thanks so much for having me.
Thanks, Heather. Richard?
So I'm Richard McFarland. I'm the Chief Regulatory Officer of the Advanced Regenerative Manufacturing Institute, which runs quite a few programs, including BioFab USA, a DoD-funded manufacturing institute. I'm an immunopathologist by training and also spent about 17 years at FDA. Maybe that's the shelf life of an FDA employee. I was always in the Center for Biologics. The office names kept changing, but first as a reviewer, back when they were doing recombinant proteins and monoclonals, as well as gene therapies and cell therapies and xeno.
Thanks, Richard. Tim?
Mike, first of all, thanks for the invitation to be here today. I appreciate your work as chairman organizing us. I'm Tim Schroeder, CEO and founder of CTI, Clinical Research Organization, that specializes in cell and gene therapy and has some xenotransplant experience. Prior to starting CTI, I was a transplant immunologist, and the saying around xenotransplant is it's been right around the corner and always will and always has been, and I think finally we have reached around that corner and looking forward to the discussion today.
Great, thank you very much. My name is Jason Wertheim. I'm a transplant surgeon at University of Arizona, the College of Medicine in Tucson. I'm also Vice Dean for Research and Graduate Studies there. And I run a lab in tissue engineering, working on liver and kidney tissue.
Thanks, Jason. With that, we'll go ahead and get started, first with an introductory presentation. So the session scope is the panel will explore the evolving landscape of xenotransplantation, which is a type of regenerative medicine, in fact, including discussion of history, technological advances for reemergence, current state of FDA regulation, recent successes, and related tissue engineering. We will also explore numerous CMC, clinical, and regulatory complexities and ways to navigate in order to solve the organ and tissue shortage problem. So why is this topic even important for this audience? Well, regenerative medicine refers to a general approach to restore, replace, or recreate cells, tissues, or organs to treat or mitigate disease. This is one of the numerous regenerative medicine definitions, but it's a good one. FDA also has a regenerative medicine therapy definition as it pertains to the product definition associated with an RMAT eligibility, and we'll get into that a little later. Both xenotransplants containing viable cells or xenografts devoid of viable cells are classical forms of regenerative medicine, and this is the Alliance for Regenerative Medicine. Some history of xenotransplantation and recent successes. So Dr. Thomas Starzl, back in the 60s, while he was also known as the father of modern allotransplantation, also performed xenotransplants between baboons and chimpanzees and humans. And although there was limited success, this paved the road for modern xenotransplantation. For numerous reasons, including physiological, based on size, and agricultural, based on scale and capacity, pigs are the primary focus for human xenotransplant donors. Some of the technological breakthroughs that got us here today include pig cloning breakthrough back in the year 2000 by a company that was also involved in cloning Dolly the sheep. And this was done via somatic cell nuclear transfer. That technology was then used in the application for the first relevant knockout for the purposes of the xenotransplantation field, which was to knock out the GAL antigen that is associated with hyperacute rejection, where rejection occurs in a very small amount of time. That was published in Science, and it was done via classical gene knockout technology like homologous recombination. The screen went dead there, okay. At that point, there was a gap, and that gap required technological innovation. Technological innovations for that gap included gene editing technologies like CRISPR, efficiencies and refinements in the somatic cell nuclear transfer process, new science, a better understanding of zoonotic or cross-species infections, and other reasons. But in 2017, another advancement. The PERV, or porcine endogenous retroviral vector, that's known to be, at least perceived to be, a zoonotic agent risk, has 62 copies of it in the pig genome, and therefore required a technology like CRISPR in order to knock all of those sites out. That was published in Science. In 2022, another group published on a 10-gene edit pig, which was a combination of knockouts and transgenes known to be important, and performed the first pig-to-human heart transplant under compassionate use IND, which was published in 2022 by the New England Journal of Medicine. More recently, at least 3 different intentional genomic alteration, or IGA, pig sources have been used in either a decedent model, which is a model based on a brain-dead human under informed consent, and or a compassionate use or single patient IND, and some of which have been published in the New England Journal of Medicine over the last few years. These were mostly with kidney or thymokidney organs. This year even, there's been at least three INDs allowed to proceed for each of these IGA pig sources for human xenotransplantation using kidney or thymokidney. United Therapeutics has a press release, as well as eGenesis, on this. While this session is really focused on xenotransplantation for the most part, we would be remiss to not also discuss some of the other important areas of tissue engineering and organ engineering. Organ engineering is in late preclinical stages, including decellularization of pig organs, then recellularization using human cells via technologies such as perfusion. Other types of tissue engineering include the development of human cell-based combination products. Note that medical devices like acellularized heart valves, skin, and other organs, or other tissues, I should say, either from pig or bovine sources, have been used for years in humans, and we will discuss it briefly later in the session. For regulation guidance and terminology, the major difference between a xenograft and a xenotransplant is that the xenotransplant contains viable xenogeneic cells and is subject to CBER's xenotransplant guidance, which you can see on the lower left. Although not much has been changed in that guidance between the 2003 version and the 2016 version. Given the swift progress in the field, I expect that to change. Xenotransplants are regulated by CBER, but if sourced from an IGA animal as well, then CVM will first regulate, and there are 2 relevant guidances here, one in the lower middle and one in the lower right, published in 2024 and 2025. All of these are final guidances. Xenografts have had cells removed by acellularization or decellularization. One needs to show viable cells, and all pathogens are removed. And they are not, for the most part, subject to the CBER xenotransplant guidance, although there are lots of relevant other guidances, as well as standards. Xenotransplants often regulated by CDRH, unless combined with human cells and tissues, in which case it would be a cell-device combination product primarily regulated by CBER. Also of note, xenotransplants are likely eligible for RMAT designation. If you look at the 2025 draft guidance below, the quote is, further, as FDA interprets Section 506G, xenotransplantation products may also meet the definition of regenerative medicine therapy. And in fact, in the version prior to this, it only mentioned xenogenic cellular products, so obviously there was a purposeful change. What is on the horizon? Well, given that there's 3 INDs allowed to proceed this year, human trials under IND will kick off soon, so that's exciting. Other organs besides kidneys and thymokidneys will, of course, progress, including liver, potentially heart, and others. I think there will be a determination of what's a need to have versus nice to have from a gene editing perspective, because there's obviously technological and regulatory risks with regards to tacking on or compounding gene edits. There are ways to control xenorejection with immunosuppressive drugs that often are first tested in the allotransplant setting. It is important to note that these are CEDAR INDs because FDA does not regulate human organ allotransplantation. And it will be important to understand the utility of seamless clinical trial designs that seem to be favored currently, as well as the potential eligibility and utility of the RMAT designation. Other than immunosuppression, tolerance induction is also something that has been studied for some time and is also reemerging. Just to flash the planned session topic coverage, we're going to cover dual center regulation, some of the compassionate use or single patient IND versus commercial IND points, the benefits and risks of gene editing in IgA animals, zoonotic agents, which again is cross-species infectious agents, the supply chain when you go from an animal facility to what is more GMP-like to house a donor pig, and then to more of an OR setting from an organ procurement perspective. And then everyone's fun topic of comparability, well, it happens in xenotransplantation too. What happens when you start with one IgA pig and then decide a second generation pig needs to be implemented, and what do you do about it? And then, of course, mentioned before, the use of tissue engineering, including pig organ scaffold and human cell components as an alternative to the primary topic of the session, which is the use of IgA animals and organ xenotransplantation. Of course, we'll also cover some clinical strategy. With that, we can close the presentation, and I think Heather will be back up on the screen. Thank you. So the first topic I want to, well, actually, just before I get into that, do any of the panelists, virtual or present in person, want to add or edit anything I may have said in the slide deck?
Well, we'll get to xenografts. The one thing, there is a company, ARMI member, Miromatrix, who is in clinical trials, publicly released clinical trials for the recellularized porcine scaffolds in livers.
Okay.
So it's beyond preclinical, it's actually in early phase 1 trials.
Oh, thank you for that update. And anything else I may have missed?
Mike, I was just going to make a comment. I think from the standpoint of the field and this being so important, it's going to require a lot of really thoughtful decision making, and unfortunately or fortunately, it's going to be very high profile in the media. And as such, I think a misstep is going to be under the proverbial microscope and be very heavily scrutinized. And so that's something that many times in the area of drug development is there, but I think this is going to be even magnified maybe 1,000-fold.
Yes, I agree. Jason?
Yeah, I would say that's a really important topic there, because even with the early stage pilot studies that we've seen over the last couple of years, now moving into clinical trials, indications for those clinical trials or indications for xenotransplant from a patient selection standpoint is going to be very center specific. New England may be different than the New York centers, may be different than West Coast centers and Midwest, sometimes based upon wait times for a human organ for transplant. So those are some of the things we can get at as we move through the discussion topics.
So can I
Of course.
comment on Jason's comment? I think that's a really good point, because regardless of the source of the organ or the tissue or I was at ARPA-H 2 weeks ago, I think, and they have a program for biohybrid organs.
Yeah.
So independent of the source of the organ replacement, I think working with the oversight at HRSA, which is my understanding in a bit of a less than stable place right now, and the concept of how you oversee allo organ transplant started before we thought about cell therapies or gene therapies, right? And so it's a very different model. And how is society going to deal, as to your point, how is it going to deal with these different sources of organ replacements? Because we all know that there's never going to be sufficient allo supply chain for all the needs. And how do you do that on an individual trial basis? And how do you do that on a societal basis? I think it's going to be really important. And Xeno's going to jump right in there.
Agreed. Heather, do you have anything to add?
Yeah, I just want to comment on Tim's comment a little bit. I really appreciate what he said, regarding sort of the light in terms of the public view and, getting it right. And I think that, we're all committed to this as an industry, but also FDA is as well. And I think it really highlights the importance of engaging with the agency throughout the entire process. This is a new and exciting area, both in the cells and tissues for xenotransplantation, but also in the tissue engineering space. And I think FDA has been, in my experience, very open to having conversations with developers, both on the CVM and the CBER and CDRH side, and are sort of willing to go through this with us together to make sure we get it right.
For sure. Okay, we'll move to the Q&A portion of the panel, and of course we encourage audience participation. So let's start with what we would call the dual regulatory issue that exists for IgA animals in human xenotransplantation. I'll start with a general question. What is the regulatory process for a company interested in developing an organ for xenotransplantation for human use from an animal with intentional genomic alterations? Maybe we'll start with Heather.
Thanks, Mike. Yeah. So an intentional genomic alteration in an animal is regulated by the Center for Veterinary Medicine. So whenever you have an IgA in an animal in place, you are going to have sort of this dual regulatory system where you have approval of the CVM product, which is the IgA, and then separately you would have approval of the human therapeutic. So in terms of the regulatory process, I think it's critical to sort of, if you're approaching this as a developer, to think about what resources are available to me as a developer before I even engage with the agency on this. And you highlighted a lot of these in your presentation, Mike, but there are various guidances that are available. There are webinars. CBER even has its own website called OTP Learn that has various, even very basic webinars about the approval process. And then approval documents are also published where you can learn about other products that have gone through the approval process that may be similar. So it's really important to sort of do your research and understand, about the resources that are available. And then the next thing I'd say is really determine sort of what your strategy is, how you intend to market the product, and actually what is the product. And then thinking about some of the things you were discussing, Mike, who's the regulatory center that would have oversight of the human product? So we already mentioned IgA is within CVM, but if you have a biologic like tissue or organ for transplantation, generally CBER would regulate that product. Whereas you mentioned xenografts, if it's just a xenograft alone, that would be considered a device and a CDRH product. And then you have the complicated factor of combination products where you may have like a cell scaffold, a device biologic combo. And generally, sort of there's a designation of a lead center. And that's primarily based on the primary mechanism of action of what's making that product work. And in the case of a cell scaffold, that would essentially be the cells. So generally, CBER is the lead center in that process, although they may consult CDRH for their expertise on device-related matters in the approval process. So it's important to sort of understand the logistics behind the approval process. And then, as you mentioned, the NADA, so the new animal drug application, is the CVM approval, and that has to be in place prior to the human product approval. So you would get your NADA first, and then you would seek your BLA, for example, if it's a biologic. And it has to happen sort of in that stepwise manner. Preferably, you would work with CVM very early in development of that IgA in the animal, and you would sort of go through that process prior to initiating your clinical trials. Now, there's no hard and fast rule that says that you have to do it in a certain time frame or you have to do it, way before you engage with CBER. The only rule is the NADA has to be in place before the BLA, for example, can be approved. So you can even go through the process simultaneously. But I think it makes a lot of sense to engage with CVM and really work on, development of that IgA in the animal, because that's really what's coming first in the process. And then obviously you would also need to engage with CBER early on, just to let them know sort of, this is the product we have coming up, and also to understand what the regulatory requirements are that you'll need to go through and have in place prior to the BLA approval. I will say that usually all of the centers have great programs to facilitate this early engagement with the agency. CVM has what's called the Veterinary Innovation Program, which allows for enhanced communication and other resources like pre-review feedback. CBER has various resources, including, you mentioned RMAT, so the RMAT designation. There's also the ability to engage early in what's called interact meetings, which used to be a pre-IND meeting. So you can have those early meetings during development and get your questions answered early by the agency.
Thank you, Heather. Richard, did you want to add something?
So as Heather was talking, there are a couple points. One is that the different centers at FDA do talk to each other, but they don't necessarily know that you've gone to CVM. CBER may not know you've gone to CVM until you tell them, and then help make that connection. In some ways, I see it's sort of a sequential process. In CVM, early discussions would be analogous to making, if you were doing something that was a human cell containing thing, you make sure that you collect your samples following the tissue rules. And then it becomes sequential. Similarly, you'd want to get to CVM, talk early. Some of the studies that you might do for CVM could also work for something for CBER. But it's up to the sponsor to some degree to help facilitate that discussion, because the agency is happy to talk to each other. She and I both have history of talking across centers when we were at different centers, but the sponsor owns that product development and needs to kind of help the agency make that connection, because we don't share everything just to share everything. There's just too much going on to do that.
Very true. And I'll just add, again, given that the NADA is required before a BLA, but that doesn't mean that an INAD is required before an IND, although there are significant advantages to doing so, and there are, I guess, a plethora of disadvantages, potential disadvantages. As Richard said, you might lose some efficiencies by not knowing what CVM needs versus what CBER needs, for example, or the other way around.
Yeah, and that's true with the combination. I'm sorry, that's true with combination products as well. And in some ways, Xenos are combination products in that sense, and you have lead centers, but you got to understand all the parts of your product.
Yep, and then in addition, one can ask, for example, the Interact or the pre-IND for CBM presence, although that might also just happen via CBER internally as well. We'll move on to another question. Richard, I did briefly discuss the difference between a xenotransplant and a xenograft. Did you want to give that a little more flavor from the regulatory perspective?
Sure. So you pointed out the key difference is the lack of viable animal cells in xenografts. And the presumption behind that is without the cells, and they work primarily, traditionally, they were seen to work physically. So porcine valves used in cardiac surgery is kind of the classic example of a xenograft. The more that, and those were regulated as medical devices, a bit of the rub comes in when it's a xenograft that has some biological activity that functions as well as its physical activity, and that's go to Office of Combination Products and ask a jurisdiction question about that. And related to the case I mentioned earlier about mirror matrix, that is a combination product, so it's a scaffold, and it also has some extracorporeal parts that are classical medical devices.
Right.
And so that sponsor needed to talk to multiple centers, although CBER was the lead center because that's the primary mode of action that was easily to be determined by the human cellular component.
Right.
So
great. Does anyone in the audience want to speak to anything that we've presented thus far? If not, we will move into our next topic. Okay, so the next topic is the similarities and differences, so to speak, between a compassionate use or single patient IND and a commercial IND, and also the use of what's relatively new in general, as far as I know, but definitely in the xeno field, which is the use of a brain dead or decedent human to generate potentially even better fitting preclinical data than you can in for example, a non-human primate. Start with the first question. Can you please provide some of the differences between a single patient or compassionate use IND and a commercial IND? I'd invite Jason or Tim to start.
Yeah, so I think, when we were seeing the very early stages, even a couple of years ago or so, in xenotransplantation, particularly with transplantation of the heart as an example, or even early kidney transplantation, those were typically compassionate use in the context where patients were at a very end stage with no medical treatment in immediate future for them. As an example, maybe a patient has lost dialysis access, as an example, or end-stage cardiac function. In that case, for clinical care, compassionate use can provide a potential therapy. Then as we go now more towards clinical trials, those clinical trials are now looking at efficacy, even before that safety, and trying to look at how well do the xenotransplants function within the person over a population of 10 or 20 or so sample size. And that provides specific data on efficacy, as well as trying to help out with immunosuppression. The decedent models, and we've seen a couple in the medical journals and in the press, where a kidney transplant was implanted into a deceased individual under IRB authorization and consent. Sometimes those have been implanted for several weeks. That allows the investigating center to do physiology studies, very closely monitor the function of the graft, perhaps do intermittent biopsies if need be. There's less of a risk there for bleeding and injuring the graft, as an example, compared to if it was in a living person. And so that allows also the testing of different regimens of immunosuppression, and also potentially even different flavors, so to speak, of organs with different mutations, let's say in the complement cascade as an example, to try to reduce rejection. So that's one of the benefits there of the decedent model, and the decedent models are fairly recent in the last several years, right, having the ability of these scientific investigations in that aspect.
Yeah, so I think it definitely helped describe some of the benefits of the human decedent model. I guess we should point out specifically, though, that and you mentioned correctly, there's IRB and informed consent involved, but FDA does not have specific oversight. I don't know if Heather or Richard, you want to chime in?
So I just want to ask Jason a question. How closely does the decedent model, the IRB studies, relate to what a transplant center might have under IRB for allotransplant? Are they similar sort of ideas as you try, as a transplant surgeon, as you try to improve that process? You try to use allo organs with better acceptance, and are they similar in that conceptually?
In a matter of speaking, yes. So blood compatibility is important in all the different contexts there, as well as cross-match. So in clinical transplantation, we have two ways or two pathways to look at organ and recipient matching, for instance. Number one is matching the blood group, and then number two is looking at HLA matching, and that's dependent upon basically a cross-match. And the pigs, there's porcine flavors of HLA, and they represent another obstacle in terms of rejection, and there's some overlap or cross-reactivity between the two. So in some sense, yes, there's similar perspectives that we're looking at in terms of the decedent model. But really, for the decedent model is the fidelity of being able to do frequent sampling. So, we probably, after transplant, bring a patient in for a blood test, maybe every 3 days, once a week, as an example, for standard of care clinical transplantation. But in a decedent model, it's possible to look at urine compositions on a daily basis, or even on a every 6 hours, and fluctuations. In particular, looking at in a kidney transplant, looking at the creatinine, as that may suggest rejection. And the creatinine usually is a lagging indicator, unfortunately, of rejection, and there's different ways of trying to diagnose that earlier.
As I may remember, it's a lagging indicator, and it's a non-specific indicator as well.
Exactly,
right?
Yes. So it could lend itself to either rejection, or sometimes an infection, or another times just dehydration, essentially, lack of decreased perfusion to the graft.
Mike, just a quick comment to Jason's points. You know, for the audience's benefit, the families of deceased donors are incredibly altruistic. At the worst times in their life, they're being asked to donate their loved ones' organs, and yet they do that on a repeated basis. I think this setting actually is even a next level up. If you think of it, they're not able to move forward in many cases with funerals and things like that for several weeks,
Right.
purely for the good of us from a science standpoint. And I don't have an answer to this, but you know, for many of us, the issue of compassionate uses in the drug development of other therapies, it's there. But I would say I don't know if it really plays a major role in drug development. I have a feeling that the compassionate use of the decedent model as we move forward in thinking about this as a field is probably going to still be front and center, because it does allow you to do some things that we can't do in prospective randomized trials. I don't know if you agree with that, Jason.
Yeah, that's correct. And so, you know, there's been the decedent model looking at kidney xenotransplantation, liver xenotransplantation as well, and really trying to kind of get some of that early data that then will help us manage the patients down the line once there's a you know, human transplantation. It informs a little bit on immunosuppression. It informs on suspicion of rejection, because sometimes doing a biopsy, there's a risk of bleeding. So there's always a risk and a benefit there. One thing I want to say, though, and to your point, is that the families are very altruistic, the patients as well, too, in that you know, they're really looking to advance medicine and technology.
Thank you. Let's move on to some other topics. So sort of a clinical strategy question, but I want to reiterate the question that I asked in the last topic. So please speak to patients participating in these early trials and specific considerations around them. And then if you can, I'll start with Tim and then to Heather. Highlight for me what might look a little different in the compassionate use or a single patient IND setting versus the commercial IND setting.
Yeah, I guess to start with, Mike, and far be it, I mean, we've got some amazingly talented, smart, gifted people on the sponsor side at United Therapeutics, at eGenesis, at Micromatrix, at the clinical side. I don't have any concerns that we're going to find our way through the path to do multi-center randomized trials to prove the efficacy of xenotransplant and where it places relative to all the other treatment options that Jason and others will have. The concern I have, and concern might be a strong word, but who's representing the patients here? In that, it's really hard for us to put ourselves in a position of a patient and a patient family where all of the things going on with end-stage organ failure, and as you described, Jason, if it's a kidney transplant or end-stage renal patient who suddenly loses vascular access, or if it's a heart or liver failure patient who suddenly has no treatment, and suddenly to say, we're offering you the opportunity to go into this controlled study potentially, I just wonder, you know, relative to which patient population is the appropriate ones to be in here. How do we truly get informed consent? How do we decide which centers? Because there are probably just a handful of centers that should be doing this research at least to start with. How do we decide once they've consented to be in the study, you know, there'll be a protocol that they have to follow, but I think the issue of what if I want to opt out as a research subject or as a family member, the complexities of this on the patient side, I just don't think can be underestimated. And it doesn't mean that people like Heather and Jeff and people like that, Mike at eGenesis, aren't thinking about this. But I think it's really hard for us to put ourselves in the shoes of the patient and their family, who ultimately, when this is successful, the subsequent generations will benefit. But this initial cohort, I think it's going to be really tough.
Agreed. Care must definitely be taken.
Right, that's a good point. So as an example, for standard of care human transplants, for a kidney, the wait time in California is probably over 6 years, probably over 8 years. Similar kind of wait times in Chicago, where I used to work, in New England as an example. In Arizona, it's about 3 years, just as a comparison. So sometimes we may think of xenotransplantation as being then a bridge to human transplantation. So as an example, so one indication could be a patient that's maybe been 2 years, 3 years on the wait list, but then has another 5 years to wait for a human transplant. Is that the right time to intervene on a xenotransplant, as an example? Or is it a patient that is highly sensitized? And so what do I mean by highly sensitized? So I talked a little bit earlier about HLA. Well, humans can have anti-HLA antibodies, either if they've had a blood transfusion or a prior pregnancy or a prior transplant for that matter, and have pre-formed antibodies against other donors, human donors, out in the community. Well, if someone has complete sensitization, which is what we call a panoreactive antibody, a PRA of about 99%, is that the right patient population then to transplant with a xenograft? Again, I think we're still trying to figure out the sweet spot now for the clinical trials. And again, that's different conversation than one would have for, say, compassionate use that we spoke about earlier.
Yeah. Heather, do you want to weigh in?
Yeah, a little bit of a different direction, but speaking from a regulatory perspective, I'll just make the general comment. We were talking about decedent studies and now single patient INDs. I really think it's been great for Xeno and sort of moving the needle and making progress in this space because of all the challenges, I think, with animal models and particularly NHP studies. You mentioned that FDA doesn't have oversight of decedent studies, which is correct, but I still think they can provide valuable information in terms of proof of concept and some safety data. In terms of, from a regulatory perspective, the key differences between a single patient or what we're calling compassionate use IND versus more of a commercial IND, I mean, it really boils down to the risk benefit calculation. So the benefits of the therapy need to outweigh the potential risks. And this is particularly important in a single patient IND setting. And I think there's a little bit of a misperception that there's a lower bar in terms of regulatory requirements for single patient INDs. And I don't think that's necessarily accurate. It all goes to that risk benefit calculation. And a lot of times these patients that are participating in these studies often have severe debilitating conditions, such as what was mentioned in end-stage kidney or end-stage heart failure, and they really have no other options. And so it's a very unique case in these single patient INDs, and the treatment is really up to the discretion of the physician. Whereas with the commercial IND, it's a bit more of a controlled setting. You have multiple subjects that are participating under a single defined protocol with defined inclusion and exclusion criteria that would be unique to that study that wouldn't necessarily apply in the single patient IND setting. And it's thought that maybe you have increased data requirements for the commercial IND. I think that's partly true, but that's because of that safety component. A lot of times these patients aren't sort of in the same debilitating condition that maybe the patient in the single patient IND setting is in. So it's just a different scenario, and the data requirements are therefore different.
Fair enough. Maybe we can get a little bit into what are the pros and cons of certain trial designs. So both press released, INDs allowed to proceed, speak to a seamless phase 1, 2, 3 clinical trial design. And just curious about thoughts around the panel. I know, Heather, you have obviously directly relevant experience with the United Therapeutics IND. What are some of the pros and cons?
Yeah, sorry, trying to come off mute. I think it's an exciting time. I will say these sort of phaseless trials, it's not really unique to Xeno. We see it in other settings, such as oncology. But it is sort of a nice, like flexible design that I think is being utilized for Xeno trials. Some of the pros is, you're combining your phases and it allows you to sort of build in safety into the initial phase. And then typically it's more of a cautious approach in the beginning where you have sort of a smaller number of subjects and then you're enrolling maybe more patients as you progress through the study into sort of the later stages. Typically, the agency will define sort of a ceiling in terms of the number of patients required for that study. The hope is if the data look good, then maybe you don't necessarily need to hit that ceiling. But again, I think that is going to basically involve conversations with the agency, to make sure everyone's on the same page in terms of what the data is looking like over time and what would be required for the BLA. Some of the cons of this approach, I mean, it can take a while to complete the study, especially as I said, you're sort of doing it in this paced manner. Usually it's just a small number of patients in the beginning before you can sort of increase the patients within the study. There's no guarantees from the agency that what they're defining in terms of the number of subjects will be suitable to support a BLA. It's a lot of unknowns at this point because I think we're still sort of in the early phases of this, but I definitely think it's exciting times. And like I said, the agency seems more than willing to work with us on this and are really open to that increased communication throughout the trial.
Thank you. Tim, do you want to add?
Well, I was just going to say, Mike, it's interesting, and this is not a comment on either United Therapeutics or eGenesis, but we do see the phase 1/2/3 occasionally in other settings like oncology. And I would say without any statistics at my fingertips, more often than not, they don't move all the way through. What typically happens is either there's a lack of efficacy and the sponsor makes a decision not to move forward, or there's tremendous efficacy, in which case you go back to the agency and actually ask if you can combine, later stages together. But I will say, I almost think it's a stamp of, I don't know the right word, affirmation that's more important for the shareholders and maybe the venture capital community, the investors, to say there is a path to get a xenotransplant approved as shown by FDA. And I think that to me is probably the more powerful thing than do the companies actually get a chance to go all the way through it. But that's just one person's opinion. But I don't know, Richard, you sit in the FDA seat, or obviously Heather, you did before, but.
So, yeah, well, we were both at FDA, and people in the audience were at FDA and dealt with FDA. Even in biologics that aren't xeno, you're talking about phase 1/2. That's a recognition of the reality of products that are developing or developed differently than small molecule drugs, which was when that paradigm got in everybody's head. Implantable medical devices really just have a exploratory trial and a confirmatory trial. And I think, so in some ways, a 1/2/3 is a blending of that, and the regulatory expectation is that it's reasonable scientific evidence. It shouldn't be written in stone. It's what's best for the patients and how do you get there. And I was going to ask, I keep thinking about what happened at the birth of allotransplant, where it in some ways is very similar. We're dealing with patients who are being participants in trials where there is no other option, similarly to oncology, similarly to Emily Whitehead in CAR-T. She had already failed transplants. And in that case, being able to show that this alternative versus standard of care, you have a much larger delta than if you're trying to figure out the 17th thing for mild headaches.
Thank you.
Well, I mean, I think that, it's an exciting time, from many different perspectives. And, whenever we can offer a new treatment to a patient, it's certainly eye-opening and really provides hope. The wait list for organ transplants typically goes up, and up. It's always a supply and demand question, and there's not much more supply of human organs, unfortunately, really encouraging living donation as an example, but still we've reached a plateau for those organs that we can transplant from living donors. And so we've been looking for alternatives, and actually, so xenotransplant is a new hope for those patients who sometimes are facing years on a wait list. Or debilitating diseases in the context of chronic liver failure, as an example, or a heart failure.
Death.
And death, exactly. So it's really an exciting time. And yes, there are some similarities going back and a lot of breakthroughs. And so, as many of us have seen on the news, the Nobel Prize this morning was awarded for regulatory T cells, which is kind of the basic building blocks of tolerance, and potentially one day we may never need to use immunosuppression, either for human transplants or xenotransplants. And so there's a lot of hope out there.
Very
There's
good
a lot
point.
of work too, but that's why we have to do the studies. And
You keep tempting me with immunology,
Very good point.
Jason.
So just wanted to, again, ask the audience if anyone had any questions for the panelists or wanted to speak to a comment. Please go ahead, Larissa.
Hi, thank you all very much for your comments. I wanted to go back, partly crystal ball, and get you guys to the crystal ball, and Heather, you too, I don't know, about the role of xenotransplant organs as a bridge to further allotransplants or to Richard's regenerative organs.
Yeah, I'll just repeat the question.
Use the microphone there, so
or maybe, yeah, with the
make sure
microphone.
Heather
please.
can hear.
Hi. I'm sorry, Heather, if you didn't hear. I was asking all of you if you would be willing to crystal ball into the future as to the role of xenotransplants as either one, bridging organs for very ill patients who need to get better before they're ready for an allotransplant, and two, as bridging organs for the regenerative organs that Richard's group are going to be making.
Great question.
yeah, so just to start that conversation off, I think that you're talking about a bridging therapy versus a destination therapy, and so right now, human transplantation standard of care would be our destination therapy, and so for many organs, several organs, it's based on wait time. For other organs, actually what determines someone to get a transplant is how ill they are, for lack of a better term. And so at least in the early stages, what some places are doing for kidney is utilizing xenotransplant as a bridge to a later human transplant where a patient would not want to be on 3 more years of dialysis. But if they could get 3 years of function out of a xenotransplant and then go to a human transplant, that's a possibility. So in many times when someone is getting a xenotransplant, they will continue to accrue time on the kidney transplant list. So that's one particular benefit and a model as well. For engineered organs, there's a possibility, and again, this is really in the future preclinical studies, of utilizing induced pluripotent stem cells, or iPSCs. So one day actually using autologous cells to build an organ as a destination therapy. Still in the very early studies, but possibilities that are out there that, again, provide hope.
Right.
One population that's intriguing to me, to expand on the outside the go extra-renal in the conversation for a second, is used to be, and I assume this is still the case in the clinic, where it's pretty common at a transplant center that a few times a year you would get a young, otherwise healthy individual, typically female with menstrual pains, who had binge alcohol on top of Tylenol and ended up in acute fulminant failure. And either they recovered, or you did a liver transplant, or they died. Those were the three options. And in one hand, the ability to pull a liver out of the hat and be able to transplant them very quickly, you sometimes got lucky and sometimes you didn't. But suddenly you had this young individual who woke up a few days later and you're told, oh, you just got a liver transplant and you're on lifelong immunosuppression. And invariably, they typically were in the demographic where compliance and acceptability weren't high. I think that's a group where, to your point, just could be really, ripe where, you could find out, are they going to recover? If so, then you can make the decision, do we take the porcine organ out? And if not, then the issue becomes, well, we can keep it in long-term, and or we could look at other engineered approaches.
Yeah, so I usually tell my patients that a transplant is like a jigsaw puzzle. There's all these different pieces that we have to put together. And so what are the patient characteristics? What are the donor characteristics? How long have they been waiting? What is the heart function, as an example? And so, to your point, for liver transplantation, for liver failure, we don't have a standard of care dialysis, per se, for the liver. And so that is a particular area for an engineered organ, as an example, or even a xenotransplant, to provide a specific bridge then to a transplant.
Agreed. Just because we only have 4 minutes left, I wanted to see if there was any other audience questions or comments. Yes, please go ahead. Can someone please bring the microphone?
I'll be there in just a second. So as the microphone comes down, I just want to say, immunology, second transplants, whether they be xeno or allo, gets very complex very quickly, and it's something that needs to be considered. You have a microphone. Question.
Thank you. Yeah, this is a great discussion. I'm Sophie Wei, the co-founder CEO of Xtherma. So we also work on the organ transplant field by innovating in the biopreservation field. So have FDA breakthrough designation by turning kidney transplant window from less than a day to potentially 5 days. Here, actually, I wanted to ask the panelists, so if we look into the future, when xenotransplant become the standard care in the future, so what are some scaling up challenges do you envision to face, and what's some strategies?
Yeah, I'll just comment initially. A herd size of 10 versus 100 versus 1,000 is of course different. And then, as we were alluding to earlier on, we won't have time to get into today, is sort of that supply chain going from that to something that's more GMP-like for the specific donor pig, and then something that can be more in an organ procurement setting. So scale is important, geographical location and whatnot, obviously, it still holds challenges therein still hold true, whether you're talking about a xeno organ or an allo organ. But go ahead, please.
Yeah, I'd say that, we had a very good discussion earlier at the 11:30 hour, where they were talking about the logistics of CAR T cells and how to do the cell preparations and get them to patients either in the larger cities or in the community. In the future, I think we're going to encounter something very similar in the context of xenotransplantation, is how to get the organs from the herd or a GMP facility to the hospital. Even in some areas for human transplantation, we're seeing the recoveries being performed sometimes at a central location. So that's an area of scale. That's something that the companies have been looking at doing. And I think in terms of preservation, not for human transplantation, that's certainly very important. It helps with the logistics of getting the organ from point A to point B. and I think is also going to have an impact in xenotransplantation as well, too.
Thank you. We only have a minute left. I just wanted to mention a couple of quick things, and then I'm going to pass it to the panelists for a few seconds each. But a couple of questions we didn't get to is it required to have a PERV negative pig as a source for human xenotransplantation? And the short answer is no, but very much worth further discussion. And then, of course, another discussion point would be related to how RMAT might be able to play a role in expediting IgA pig-based human xenotransplantation, another topic for another day. I'll pass it to each of you, maybe start with Heather for a few seconds before we close.
Sure. First of all, this was a very productive discussion. I learned a lot from my colleagues as well as the audience, so thank you very much for this opportunity. I think this is a very important conversation. It shows, how much there is still to learn in this field. It sort of shows the progress of, how far we've come in somewhat of a short period of time.
Yep.
I think it's a really exciting place to be. And, now we're thinking even beyond just xeno organs for transplantation. We're seeing development of these cell scaffolds, for example. Jason mentioned iPSCs. There's all of these different mitigation strategies in terms of reducing immunorejection in patients. Mm-hmm. There's also a lot of studies even still to be done looking at controlling things like adventitious agents and even understanding what are the adventitious agents of actual clinical concern, yes. Meaning they could potentially be latent or, there's a lot that's unknown about even transmission of adventitious agents, right. From a porcine to a human. So I think this importantly highlights how far we've come, but how much work there is still left to do, and I'm excited about it.
110%. We are over time, but a few more seconds from Richard, Tim, and Jason.
Uncharacteristically short for me. I'm really excited that this session exists at the MESA meeting and not amongst a transplant meeting, totally a xenotransplant meeting only.
Totally agree.
Great point. I'm just going to say the last question from the audience was a really good question. I think there are so many lessons learned for Xeno from CAR-T, and we've got to make sure that those lessons are somehow promulgated forward.
Agreed. Jason, last words?
Yeah, I'm just going to close by saying I think this brings a lot of hope, not just to the patients, but to really the community in transplantation. Really terrific to be here with all of you, and I think we've kind of touched on a little bit that this also develops a community of different industrial partnerships, whether or not it's preservation, organ procurement, logistics as an example. So yep. I think the future is bright, and it's really great to bring everybody here for this discussion. So thank you.
Agreed. Thanks again, everyone, for your participation, and we hope to do more of these sessions. Take care, everyone.